All right, welcome back, everyone. This is the fifth lecture in our series on spatial epidemiology, all based on the fantastic work of Professor Olin Roger Lowell. Today, we're going to get into something really powerful, Bayesian disease mapping. Now, what is that? Well, it's basically a set of techniques that lets us take messy, sometimes incomplete public health data and turn it into maps that are actually clear and, well, actionable. Super important stuff.
So, here's our game plan for today. First, we'll get our heads around the Bayesian way of thinking. It's a whole philosophy, really. Then we'll talk about why this matters so much for making good maps. We'll break down how these models are built layer by layer, look at the cool ways they capture those spatial patterns we've been talking about, and then, we got to wrap up with a healthy dose of caution.
All right, let's start at the beginning. Before we even get to the maps or the models, we have to grasp the core idea here. Think of it as a formal way of learning. It's about updating what you believe to be true when you get new evidence. It's really that simple at its heart.
So, this really gets us to the heart of the problem, doesn't it? Let's make this real. Imagine you're a public health officer in a local government area in, say, rural Nigeria. You're tasked with mapping cases of something pretty rare, like Lassa fever. Now, in a place with a small population, what happens if you get just one or two cases? Suddenly, on paper, your disease rate goes through the roof. It looks like a major hotspot. Meanwhile, the district next door has 0 cases and looks perfectly safe. But is that real? Or is it just random noise? How do you see the real pattern through all that statistical static? That is the exact challenge these Bayesian methods are designed to solve.
Okay, so how does it work? Well, the whole process kicks off with something called a prior distribution. Don't let the term scare you. It's just a formal way of stating our initial belief. It's our educated guess before we even look at the new data. You can think of it as everything we already know, maybe from past research or historical trends or even just expert opinion. It's our starting point.
Right, so you have your starting belief, your prior. Now we introduce the new evidence. This is where the likelihood function comes in. This step is all about the data. It basically asks a simple question. Okay, given what we thought was true, how likely is it that we'd see the actual data that we just went out and collected? This is where reality gets to confront our initial assumptions.
And then, this is where the magic happens. We combine our prior belief with the likelihood of our new data, and what comes out the other side is the posterior distribution. This is the big payoff. It's our updated, more informed belief. It's a smart, sophisticated blend of what we knew before and what the new data is telling us. And the key thing here is it's not just one single answer. It's a whole distribution of possibilities, which gives us a much richer understanding of the uncertainty and a way more nuanced picture of the actual disease risk.
Okay, that's the theory. It sounds pretty neat, right? But the real question is, how does all this help us make better disease maps out in the real world, where things are messy?
Oh, the advantages are huge. Seriously, remember our example of a rural district with a tiny population? Bayesian methods are perfect for that. They help stabilize those crazy high or low risk estimates, smoothing them out to something more believable. But the real game changer is this idea of borrowing strength. It's built on a simple truth. Things that are close together are usually more related than things that are far apart. So, a small area with very little data can literally borrow information from its neighbors to get a much more reliable estimate. It's like asking your neighbors for a cup of sugar, but for data. This makes the entire map way more stable and realistic.
And this quote just nails it. The whole point is to create more stable risk estimates. We're trying to see the real, underlying pattern of the disease, not just the random ups and downs that come from having noisy data. Think of it like tuning a radio. We're trying to dial in the clear signal of the disease process and filter out all the static.
So how do we actually build one of these things? Well, the framework that makes all this possible is called a Bayesian Hierarchical Model, or BHM for short. The best way to think about it is like a multi-layered cake or a set of building blocks. It's a really smart way to structure our analysis and keep all the different pieces organized.
It basically works in 3 levels. Okay, level 1 is the data model. That's the easy part. It's just where we describe our raw data, the actual number of disease cases we counted in each area. Then, level 2 is the process model. This is where we lay out our theory for what's driving those counts, the underlying risk factors and spatial patterns. And finally, level 3 is the parameter model. This is where we plug in our priors, our initial beliefs about all the unknown pieces of the puzzle. This layered structure is what's so brilliant because it lets information flow up and down the hierarchy and, crucially, between neighboring areas.
Okay, so let's get into the real engine room of these models. We've talked about borrowing strength and spatial patterns, but how do we actually tell the model that geography matters? How do we code that idea into the math? Well, that's where some very clever spatial models come in.
The whole thing really starts with what are called conditional autoregressive models, or CAR models. And the concept behind them is so simple, it's beautiful. It just assumes that the risk in any given area is influenced by the risk in its immediate neighbors, the ones it shares a border with. And that's it. It's just a mathematical way of saying, hey, neighborhoods are important.
Then, in 1991, things got even more sophisticated. A landmark model called the Besag-York-Mollié model, we just call it the BYM model, came along and made a really crucial breakthrough. The creators realized that the risk in an area wasn't just one big blob of spatial effect. No, it was actually made up of two distinct components, and the genius of their model was that it could pull them apart. So what are these two components? Well, first you have the structured spatial variation. This is the stuff that clusters, the part of the risk that's shared with neighbors and spreads across the map. Think of it as the regional trend. But then you have the unstructured heterogeneity. This is the exact opposite. It's the random, area-specific noise, the little quirks and local factors that make one area different from its neighbors for no obvious reason. And being able to separate these two was a total game changer. Why? Because now you can look at a hotspot and ask a really important question. Is this high risk because it's part of a bigger regional cluster, or is there something uniquely weird and local going on right here that we need to investigate?
And it all boils down to this beautifully simple formula. It says the log of the relative risk in some area I is just three things added together. First, you've got mu, which is just the average baseline risk for the entire map. Then you add ui, that's the structured part, the influence from the neighbors. And finally, you add vi, which is that little bit of random, unstructured noise unique to that area. And don't get hung up on the log part, that's just a standard trick to keep the math working smoothly, and make sure risk estimates don't drop below zero. The bottom line is this formula elegantly pulls apart the shared risk from the unique risk for every single place on our map.
Okay, now I've been talking up how powerful these models are, and they really are, but, and this is a big but, they are not a magic wand. It is absolutely critical for anyone using these methods to be aware of their limitations and the challenges that come with them. First off, these are not push-button analyses. They can be incredibly demanding on your computer, sometimes taking hours or even days to run. Also, that prior belief we talked about at the beginning, your choice there can really influence the results, so you have to be super careful and able to justify why you chose it. Then there's the big one, the risk of over-smoothing. You can get so focused on cleaning up the noise that you actually erase a real important local cluster. You smooth it right off the map. And of course, all of this means you really need to know what you're doing. This is specialized work. And really, that leaves us with the fundamental tension at the heart of all this work. The whole point of these powerful methods is to smooth away the noise so we can see the signal more clearly. But we always have to be asking ourselves that tough question. When does our attempt to smooth the data become hiding the truth? It's a balancing act, and it's one we have to get right. Thank you for tuning into this part of our series on Professor Lawal's work.